Okay, this is part 2 of chapter 6, and we're just going to go through mitosis. So just the specific stages that happen in mitosis. And in this slide we have at the top a cell in interphase. And we've talked about interphase as part of the cell cycle. So the cell functioned as just a cell. And during interphase, if it got the appropriate signals, then that cell copied its DNA. So we can see that this cell already has 2 strands of each of the colors, blue, green, and orange. So it's already copied its DNA during interphase. After it copies its DNA, it moves into mitosis. Now, mitosis specifically refers to division of the nucleus. It doesn't include anything that happens in the cytoplasm. So mitosis is only focused on the nucleus, what's happening with the DNA. And the first step there, so chromosomes condense, so very distinct structures. It's tightly wrapped up. And that's very important for cells because the amount of DNA inside every single one of the average cells, if you blew that nucleus up to about the size of a tennis ball, the amount of DNA inside there would be the equivalent of 24 miles worth of fine string. Now, in humans, where we have 46 chromosomes, that would mean we have 46 pieces of string. If you lay them end to end, they would add up to be miles and miles, 24 of them worth of DNA. And you want to make sure that those are tightly wrapped up and packaged so that they don't get broken or damaged in any way. And so they get very, very tightly packed into those chromosome structures. And that takes some time to accomplish. So the chromosomes condense. That's the first part of mitosis. And we'll talk about some other details. Once they're condensed, then they'll line up in the middle of the cell to make it easier to separate them. 2 identical halves will split, and then they'll collect into 2 separate cells. So we'll go through each of those steps, and I'll make sure that you have notes to write those down. This picture over here is just the cutest thing. It's actually showing cells that are just about finished with mitosis. So they're still slightly connected here at the middle. And we'll talk about who those cells came from a little bit later on in the chapter when we get there. So starting off with interphase, it's before mitosis, but we still need to have a very important step. We need to copy the chromosomes. So we have our single strand of DNA here, and it needs to replicate. And we've seen this before. Now we have 2 strands that are held together by a centromere. The terminology for these would be sister chromatids. So these 2 are sister chromatids. And that will become more important later on, and we'll review that as we go through the chapter. But now we have 3 chromosomes in the cell, and each of those is attached to an identical sister chromatid. So we have 2 blue sisters that are exactly identical, 2 green, and 2 orange. And those sister chromatids are exactly the same. And another thing in this picture, here is an actual cell that's going through interphase. And if you look all this green stuff around here, that's actually spindle fibers that are in the cell, or different connective fibers inside the cell. And we talked about those in chapter 3. And the nucleus is, let's see, we'll change colors. The nucleus is right here. It's this blue area in the middle, and that's the nucleus. And it doesn't look really interesting. It's just a blue blob in the middle. And the reason it doesn't look interesting is those chromosomes are still all stretched out, and so it has this uniform appearance. Not real interesting. As we go through mitosis and also later on meiosis, I'm going to use this chart here, and we'll keep adding to it. So we can see for now we're starting with mitosis, and it has interphase that precedes it, followed by when the nucleus actually divides, so that's mitosis, where it divides. It has prophase, metaphase, anaphase, and telophase are all part of the nuclear division, so that's where actual mitosis occurs. And then after mitosis, we have cytokinesis to actually separate the rest of the cytoplasm, so divide the cytoplasm so that we have two individual cells. The most important part of this chapter would probably be to make sure that you get these in the correct order. A really good way to remember them would just be IP, MAT, C. So IP, MAT, C is what I've always, makes no sense, but that's what I've always used to remember them since I first started taking biology years and years ago. Other students I've had come in my classes have other really clever ways to remember this. One year I had a gal who was, a single mom. She had 3 kids working full-time and also taking a full load of classes. And her way to remember this was, I probably made a terrible mistake taking this many classes. And she did very well in the class, in all of them actually. And that's how she remembered it. So think of something that you can use to remember the order. So IP, MAT, C, and the PMAT portion of that is the actual mitosis as we go through it there, or division of the nucleus. So our first step in cell division, we have to copy our DNA, and we already know that that occurs during interphase. So we copy our DNA. That's the first step. We're familiar with that, so we know about semi-conservative replication. All of our A's and T's are together. All of our C's and G's are together. That allows us to not make any mistakes. So once we get those 2 copies completed, we'll have identical blues, identical greens, and identical oranges. So now we've copied our DNA inside the cell during interphase. After interphase, we actually move into mitosis. The first step in mitosis is prophase. And during prophase, we already have our sister chromatids copied, and they're going to start wrapping up. So we can see our chromatids are wrapping up, and it takes a while. It's a bit of a process until they form this highly condensed structure of the chromosome. We can see that as well if we look at our actual photograph of a cell dividing. It's starting to look kind of clumpy in the center of that nucleus, and that clumpiness is those chromosomes condensing. So we're going to start to be able to see those a little bit better. Here's kind of a cartoon image of what's occurring right now in our cell. And the 3 main things that are happening in our prophase, we have our chromosomes condensing or wrapping up. Also, you notice the nucleus here is starting to disintegrate. The nucleus actually falls apart. So as the DNA wraps up tightly, that wrapped up structure protects the DNA, and so it's okay to let that loose into the cytoplasm because it's tightly wrapped up. There's nothing that can get at the DNA to chew it up or break it down. And so that nucleus dissolves. Another thing occurring during our prophase here, we have spindle fibers. They're kind of these green fibers here in our diagram are stretching out from either end of the cell, and our centrioles here are migrating towards opposite poles and forming those spindle fibers. And we can see the spindle fibers here, they're highlighted or in this fluorescent green. In the cell, they're starting to form as well. So in our prophase, we always have 3 things that occur. For prophase, our DNA condenses, so DNA wraps up tightly to protect it, prevent it from getting broken or chewed up by enzymes. The spindle fibers form, so we start making our spindle fibers, and they're going to help direct cell division, help move our chromosomes where they need to be, and the nucleus breaks down. So that those spindle fibers can actually attach to either side of our chromosomes. We need that nucleus to get out of the way. So our spindle fibers will get in there once the nucleus breaks down. And those are the 3 steps that happen in prophase. Following prophase, we have metaphase. It's also part of mitosis. And in metaphase, and I should mention, your textbook doesn't include it, but most textbooks have what's called a pro-metaphase. And it happens in between the previous step, prophase, and what's happening now in this slide, metaphase. And during that pro-metaphase, is when the spindle fibers actually connect to our chromosomes and start pulling them towards the center. And then once they're lined up in the center, or on the equator of the cell, then it's in metaphase. But your book leaves out prometaphase, but just know that those fibers actually attach and move the chromosomes between prophase and metaphase. Once we're at metaphase, the chromosomes are on what's called the metaphase plate, or right through the center of the cell. We can see that up here at the top. We've got all of our chromosomes kind of squished or bunched in the center here, and all of our green spindle fibers going off to either end, and that's represented here in our diagram. It's important to note during mitosis metaphase, these chromosomes are single file, so we have our blue lined up right behind our green, right behind our orange. So they're sitting there single file right down the middle of this cell. That will become important later on when we talk about other processes of cell division that are not going to be sitting side by side. So we want to go back to our chart again and continue filling it in. So we have metaphase as the next stage, where our cells in metaphase, our chromosomes are lined up single file right down the center. So our chromosomes line up at the equator. So that's what happens in metaphase. Next part of mitosis would be anaphase. And in anaphase, those centromeres that have been holding our green and orange and blue chromosomes together let go and allow those chromosomes to move towards opposite ends of the cell. So you can think of it as those spindle fibers are pulling them towards opposite ends of the cell. What's actually happening is the chromosome is walking along the fiber, but we can imagine that they're pulling the chromosomes. That's a very easy concept to think of, kind of a tug-of-war with those chromosomes to separate them out. And we can see that happening here right through the middle of the cell. If you look at our metaphase, they were very squished right in the middle and they're starting to separate. And as they pull apart further, you'll even start to be able to see more of those ends of the chromosomes through the middle as they drag apart. So during anaphase, our sister chromatids let go of each other and start to separate. So we need to add that to our growing chart. So here's our anaphase right here. Remember, these are identical, so the orange and orange are exact copies of each other, same with the green and the blue. So our identical sister chromatids, they separate and move to opposite poles of the cell. So they leave the equator and migrate towards the north and south poles of our cell. Anaphase is followed by telophase, and in telophase, it's basically the reverse of our prophase. So everything that happened in prophase, take that and reverse it. So in our telophase, we're going to reform the nucleus. So we can see our nucleus is starting to wrap up again. Our spindle fibers are going to start to break down, so we lose our spindle fibers. Also, our DNA is going to start to unwind. It will start to relax back out into the very long strands. And we can see that up here in our picture as well. We're starting to group back into a nucleus. We can kind of see where that would be forming. We're starting to lose some of our spindle fibers here through the middle. And that's what happens in telophase. So we'll want to add that to our chart again. So now we have telophase added at the end. The 3 steps that happen in telophase, the nucleus reforms, just the opposite of what happens over here in prophase. Now our spindle fibers break down. Again, we're reversing what occurred in prophase, and our DNA unwinds. So now we've reversed all of the steps of prophase. And once those are complete, then we move on to cytokinesis. And cytokinesis is the division of the cytoplasm. So cytoplasm division. So cyto refers to cytoplasm, kinesis is division. So cytoplasm division is what cytokinesis does. So it's dividing out all of the mitochondria, the Golgi apparatus, the rough and smooth ER. So all of that gets divided during cytokinesis. And we're going to kind of ignore that whole process. Just know that it all happens in cytokinesis. the cells break apart. These are not quite broken apart up here, but they break apart and they form two separate daughter cells. So we need to add that to our chart. So now we've got cytokinesis, where the cytoplasm divides. We go from our telophase into two separate cells. So we started with interphase, copy the DNA. In prophase, metaphase, anaphase, and telophase, we carefully divide out the DNA by wrapping up and protecting it, forming spindle fibers that will help it move, and getting rid of the nucleus so we can attach the spindle fibers. We line up our DNA at the middle, and we separate those identical sisters, and then we reverse everything we did in prophase, so we reform the nucleus, we get rid of those spindle fibers, and we let the DNA relax. What's actually occurring with cytokinesis, for this class and for the exams, I want you to think of these as two separate processes. Cytokinesis, or dividing the cytoplasm, also takes a while, and it starts back here, so it takes a long time for that process to occur too, to separate out the rough ER, the smooth ER, the Golgi, mitochondria, all of that. But we can kind of ignore this part because it's easier to think of it right at the end of the cell cycle. So that way you don't get it confused with telophase. So make sure that if I ask you or give you a picture, something like this, and tell you, well, this is part of mitosis, what's happening, don't tell me that the cytoplasm is dividing. Tell me the nucleus is reforming, spindles are breaking down, and DNA is unwinding, because cytokinesis is not part of mitosis. So make sure you keep those two separate.